People are typing away, I see. For the last question, yes, which might have freaked you out thinking you had to write more. You only had to match which was um, essentially which was a lower motor neuron issue and which was an upper motor neuron issue. Yeah. So if all you did was match them, that was all that was asked. So you're good. Don't worry. I know how you guys are. Kind of. It was a little present because I do care about you guys. um, let's see. I've gotten to meet with just a few people yet about for the mid-semester appointment, and look over just a couple of presentations. I know I have a lot of appointments. Yes, I am recording. I know you have I have a lot of appointments coming up, so we'll be able to go over that some more, um, because that and what I've seen so far of the couple of presentations that I've looked at look pretty good. And just a friendly reminder that you should always be focusing really on the anatomy, physiology, and pathology of the organ in question and making the connection between malignant proliferation of cells and the symptoms. Um, that sometimes the tendency is just because, as especially people that are used to working as nurses and treating people, you tend to get pulled to like the screening, diagnostics, treatment part, but that for this class isn't needed. Um, so, like I said, between this week now, the 2nd to the 9th, I have lots of appointments coming up, so we can be sure to look at that. The nice, well, maybe nice thing or not, but last semester during this class, this was the point when we had to put everything from face to face to start to do it as online format. And because I didn't have like any equipment at all. In fact, I lost my computer for a little bit in the beginning. I recorded a lot of the lectures I gave on. I made a YouTube channel. So from this point on, there are actually full recorded lectures with me, um, in my dining area with the dry eraser board. So I'll give you my YouTube channel's name is just Kathy Soul, and there's a goofy like cartoon picture of me that says "Professor during the pandemic," like "Don't touch your face" kind of thing. So if you, I mean, I'll record this and still upload it like I have been doing. But if you wanted to look at it and kind of hear it in a slightly different way, you could also watch those videos because it's based on the same material. So that might be helpful, because sometimes the part of this lecture that can get confusing is the information about heart failure and the types of heart failure, which is very important, obviously, for this lectures for this week's content, including case study and exam, but it will come up again in that final assessment, that last like sort of cumulative take home test that you guys have. So just to be sure, because we're halfway through now, it's kind of crazy how fast the semester goes. It's both like really fast and really long at the same time. That next, we're coming on to the next case study 5 and exam 5, case study 6, exam 6, case study 7, exam 7, and that last class, just to kind of keep reminding you guys what's coming up. The last class, July 23rd, the first hour and a half will be the same like we've been doing. You do an exam and it'll be on the content we talked about the class before, which is renal function and some endocrine function. And then what will happen is we will all be presenters, and I will just sort of pick names randomly, and you will give your cancer presentation to the group, and we'll all kind of listen along. And remember that the presentation timing is 10 minutes tops, preferably less than 10 minutes as long as you get all the information across. The timer goes off. I actually time it on my phone for 10 minutes, and you do lose points for going too long. So there's nothing extra for doing extra. Ms. Also, and then we have a cumulative too. I thought yes. So you did. You're probably like, what? So then at the very end of our kind of course due dates, you'll see where it says final assessment. So that assessment is sort of kind of like a final cumulative exam. It's a larger case study, one patient who has both acute and chronic healthcare problems. So I will open that patient up. I might even do that this week, because the longer you have to look at it, the more sometimes people are happy about it. But you haven't had all the information necessarily to do it all. But it's a patient with an acute and chronic issues, and so you read the patient presentation like we've kind of been doing, and there'll be some specific questions about that person, trying to see that you're able to understand the interplay between multiple health problems. And that is worth a lot of points, actually. It's worth 100 points. So the cancer project, which is worth 75 points, and the final assessment, which is worth 100 points, are actually a really large portion of the point total. So it's like these case studies that we've been doing, and the exams are supposed to work you towards this last thing. John is saying, do we have the same patient? No, everyone gets the same person. It's sort of like a case study, like the case study assignments. Yes, very much like real life, which is, so then you're like, okay, how are all the pieces fitting together? So hopefully everything that we've worked on so far is preparing you for this last case study. And this is foundation really for physical assessment and pharmacology, when you have to then decide, so now that I know the pathologic basis and the clinical manifestations, I should be able to pick those up on physical exam. And then when you start to get into pharmacology and your track courses, you focus on what you're going to do to treat it. So hopefully we're laying this foundation for you. So that's what's coming up. Any questions about what's coming up? Rebecca. Sure. So it's easy to forget everything that you have to do coming up. You know, that first class where you see the syllabus and everything that's due, it's really hard to focus on everything that's actually due. It can get kind of overwhelming. So I like to remind people. So let me just open up the first slides, which are, and I'll share my screen here. Which is a review really of cardiovascular system function, focusing on the parts of the system, veins and arteries, and the way that the differences between those types of vessels and the heart, to set us up for some dysfunction that we'll talk about after that. So obviously the circulatory system's main job is to circulate blood so that the tissues can get oxygen, nutrients that they need, and get rid of waste products to get that immune components and substances that we learned about in the first lectures, as well as electrolytes and hormones to the area. We're regulating temperature by changing the way, if we dilate or constrict vessels, that affects temperature. There are two parts of the circulatory system. the pulmonary circulation, which is the vessels, the side of the heart, the right side of the heart, and the vessels that connect in order for the deoxygenated blood to get to the lungs and become oxygenated, while the systemic circulation is really the left side of the heart and into the system. So both the pulmonary and systemic circulations have a pump, either left or right side of the heart, arteries, capillaries, and veins. And our arteries are always the distributing system where we're getting the blood to the tissues for their needs. The capillaries are where the exchange takes place, and the veins are carrying things back to the heart. But they also have an essential job of collection and storage because they're our backup. We're not using our entire circulatory volume every minute. We have the ability to increase that if we need to. So we have our systems. The pressure between the two systems is a bit different. The circulatory system is a closed system with two pumps. The arterial pressure is much higher than the venous pressure because the pulmonary circulation especially does not need high pressure. It's not a very far system. It's not like I have to pump it from the heart to a far away distant place like I do if I'm in the regular system. And that's just a picture of the two systems working together. Blood vessels obviously are arteries and veins. So both arteries and veins are composed of three layers of tissue. The outer connective tissue, which is the tunica externa, the tunica media, which is the middle layer, which is a smooth muscle layer that constricts or dilates, controlling the diameter, and the tunica intima, which we've talked about because that's the thin layer of endothelial cells that are adjacent to the blood cells and the plasma. And we've talked about some of the special qualities of endothelial cells because they're part of obviously the inflammatory process as well as the hemostasis. The difference, even though arteries and veins have all three layers, is arteries and veins have different composition of all three layers. So the arteries have a thick tunica externa and a thick tunica media. They have much thicker muscle and fibrous outside. The endothelial layer is the same. However, the veins have a much thinner outer and muscle layer, and then they have the endothelial layer. So there's a lot less pressure. And that's on purpose because we want the arteries, we want that pressure to push the blood forward through the system to get to the capillary beds, but we want there to be less pressure in the veins because we want to be able to maintain a cardiac reserve. We don't want to push all the blood forward in the veins all the time. So because of this makeup of the arteries and the veins, because arteries are under a lot more pressure than veins, they handle fluid a lot differently. And so this is the first sort of physics equation that we're going to talk about. So compliance is how stretchy is the vessel itself. And a vein is much, much more compliant than an artery. Compliance equals, so C is for compliance. Compliance equals volume over pressure. And the key to this is the pressure, and this pressure means transmural pressure. So it's the pressure across this wall. So there's obviously much more pressure because there's more muscle and connective tissue on this side. So I'm not sure if you've ever talked about things in terms of direct or indirect relationships. A direct relationship means that when one thing goes up, the other thing goes up. If one thing goes down, the other thing goes down. They have a relationship that corresponds. And that's what compliance and volume have. The more volume that's placed in the vessel, if the volume is able to stay there, then there's an increase in compliance. But pressure, transmural pressure and compliance have an indirect relationship. The more pressure on the wall of the vessel, the less compliant or stretchy that vessel is. So, because of the makeup of the artery with that thick muscle and connective tissue layer and the increase in transmural pressure, it's a lot harder to stretch out the artery than it is to stretch out the vein. And we want that because that's where we keep our vascular reserve. Um, because we don't want to be running at optimal capacity all the time. We want to be able to have a reserve so that we could increase our oxygen output to the tissues if we needed to, if the oxygen demand increased. The opposite, and oftentimes compliance is looked at in relationship to elasticity. So veins are more compliant than arteries, but arteries are more elastic than veins. And elasticity has to do with how easy it bounces back to its regular shape. So a vessel that is very elastic has a lot of potential energy in its wall. So when the blood ejects a volume of when the heart ejects a volume of blood into the aorta, that volume of blood will expand the vessel, putting pressure on the walls of the artery, stretching the walls of the artery sort of like a rubber band. So now there's all this potential energy. Once what will happen is that artery will recoil. There's that will push come back together. Right here is the elastic recoil. And that is what facilitates blood pushing forward in the arterial system down the next segment of the artery. This elastic recoil, sort of like a rubber band. So an artery is like a fresh, thick, new rubber band that when you stretch it out, it's hard to stretch it out and it immediately wants to snap back. Where a vein has minimal elastic recoil, like an old stretched out rubber band that barely has doesn't really want to snap back at all. You can't even tell that you're kind of trying to move it outwards, which again helps is what is important for those particular structures. We want the arteries to have strong elastic recoil and a small compliance because we want them to accept the fluid but push it forward to get to where it needs to get to the capillaries. We want the veins to have less elastic recoil and more compliance to store blood if we need it. So veins are thin-walled and fibrous with larger diameters. There's more veins than arteries. They don't recoil after distension as quickly because they're not as elastic. And they have valves. So how do we move if the vessel itself, the vein that we're talking about, has low transmural pressure and little elastic recoil? How will we then push the blood forward if we want to increase blood return to the heart? And the answer is skeletal muscle. So this would be an example of looking at the vein that runs through the gastrocnemius muscle of the lower leg. So you have your vein, it's that thin-walled, compliant vessel that can store blood, and there's these valves so the blood is evenly distributed throughout the vein instead of pooling at the very bottom of it. So if I am just at rest, then the amount of blood that's circulating through is just fine. But if I increase activity, that means the oxygen demand of my body has increased. And so then I need to increase the amount of blood that goes to my heart so that I can increase the amount of oxygen. Yes, exactly. That's the reason. So I can increase the amount of oxygen that gets to my heart. So my skeletal muscles will act like what the tunica media does in the arteries. It will provide the transmural pressure. It'll milk the veins to push fluid forward so that I can increase return to the heart. So Amanda says ankle pumps when the patient is laid up. Yes, the ankle pumps are trying to simulate what it would be if your muscle in your if your gastrocnemius muscle would contract. Not because they're trying to increase preload or vascular return, because they're trying to decrease blood stasis in the legs so you don't get a clot. But it's the same thing. Instead of the muscle contracting, the pump, does that contraction. So the point is that even though my veins don't have a lot of elastic recoil, I can provide elastic recoil if I need it. And I will only need it when I increase activity. And when I increase activity, the skeletal muscles will provide that for me. So it's kind of a nice built-in system. Arteries are not very compliant. They have that increased transmural pressure and increased elasticity, as we said. So the next physics sort of equation that we have to look at is that the relationship between wall tension, transmural pressure, and the radius of the vessel. Wall tension becomes greater as the radius increases, which means that the more pressure I put against the wall of the vessel, the more tension I'm putting on the vessel. So, or to say it in a way that might kind of something that is similar would be if you're blowing air into a balloon. Right, Amanda, that's correct. If you're blowing air into a balloon, you're stretching the walls of the balloon, which is more pressure on the walls of the balloon. And the thicker the vessel, the more pressure that is. So we have talked about some of the determinants of blood flow, which is partially related to the characteristics of the vessel itself, whether or not it's highly elastic and not as compliant, or less elastic and very compliant. These are some of the other things that predict blood flow. The first is directionality, it says. So the way the blood is supposed to be is it's supposed to be layered. We have talked a lot about the components of the blood besides the plasma. The red blood cells are supposed to ride sort of in the outside next to the endothelial cells, and the white blood cells and platelets and clotting factors and all those other blood cell components are supposed to be in the middle, out of the peripheral circulation, because if white blood cells and platelets and clotting factors bump against the endothelial cells, sometimes they can become activated, and they can get activated before we need them. So that's how we would like the flow to be, layered, to reduce and prevent contact with some of those like clotting factors and platelets and stuff in the vessel wall. If there's something that takes away our layered blood flow, laminar blood flow, then we have more disturbed flow, and we have the potential to activate some of those central blood products. So if I had an area of atherosclerosis, or hardened vessel with a big plaque, then when the blood is flowing through, it could bounce and become more turbulent. So this is one of the reasons why having plaque in the artery can lead to clot formation, because we've made the flow more turbulent. Anything that does that could activate inflammatory factors or platelets or the clotting cascade. Blood flow is also determined by this next equation, pressure gradient over resistance. We've talked already about things in the body always moving from an area of high pressure to an area of low pressure, that expends little energy and the body is always working to equalize concentration. The same occurs with blood flow. There's always some pressure on the outside of the vessel from the muscle layer of the vessel and the external layer of the vessel. So that contributes partially to the pressure that we're talking about. The other part of the pressure that is being measured is the volume that's inside. So if here's the heart and a volume of blood has been pumped into the first segment of the aorta, there's more pressure here, considering the pressure on the vessel and the pressure of the volume of blood, than is over here. So that'll make the pressure gradient that helps push the fluid forward, not just the elastic recoil of the artery, but the normal pressure gradient between an area of high pressure and an area of low pressure that helps facilitate movement in the body in general. So blood flow has a direct relationship, pressure gradient over resistance. If pressure gradient goes up, meaning there's a higher difference between pressure in two places, then that favors blood flow even more. So blood flow, pressure gradient and blood flow have a direct relationship. Resistance is determined largely by, not just by, but mostly by the radius. So if we're talking about resistance in a blood vessel, the smaller the radius, the more resistance there is. So if I increase resistance, then I will decrease blood flow, because there's an indirect relationship between blood flow and resistance. So if the resistance on a vessel increases, the blood flow will decrease. And the greatest determinant of resistance is the radius. The smaller the radius, the more resistance there is. Viscosity partially contributes to resistance. If the viscosity increases, resistance increases. But it's radius that has the largest determination of flow. What that means is when we talk about hypertension, if the pressure in the vessel increases because of hypertension and atherosclerosis, so the radius has changed, the pressure is high because of the pressure on the vessel, which makes the radius smaller, then there's an increase in resistance, which means it's harder to get blood to flow, especially down to the capillary bed. So over time, there will be end organ damage because of hypertension, which of course we'll talk about again coming up. And then blood viscosity, which is the other determiner of resistance, is based on temperature and hematocrit. So high hematocrit increases viscosity. A low hematocrit decreases viscosity. So this did come up in that case study number 3 because the patient had a murmur. And the reason the patient had a murmur is the flow was turbulent because the viscosity changed. The viscosity in the blood was decreased because of less red blood cells. Pressure gradient. So Julie asked, what does PG stand for? And that's pressure gradient. Okay, the other thing viscosity can do besides changing resistance is it can cause friction damage and turbulence when the blood is flowing past, especially in areas of bifurcations. There can be damage to the sides of the vessel from increased viscosity, and that can lead to turbulent flow and clot formation. Sure, no problem. All right. And then velocity. So the speed of blood flow. The speed of blood flow is determined by the cross section of the vessel itself. The larger the cross section, the slower the velocity. The velocity is the slowest in the capillary bed, which is what we want because that's where all the exchange has to take place of oxygen, carbon dioxide, and nutrients and metabolic wastes. So when you're thinking about this, people might think, well, the aorta is like this and the capillary is like that. So wouldn't the aorta have the slower velocity? But the aorta is just one vessel, whereas the capillary bed is is the actual cross-sectional area of the capillary bed is huge because it's a large bed, and that's what you have to add it all together, not just one capillary. So from here, we will start to talk about the mechanisms and maintenance of blood pressure. We'll just take a quick break till 7:45 just to kind of keep us fresh. We're going to start with local regulation, just the short term, what's going on in this environment, which is a review because that's part of the inflammatory process, and then some of the more long term factors that regulate blood pressure. So it's about 7:36. We'll reconvene at 7:45. So I will see you then. Okay, I am back. And let me share my screen again. We will go on to discuss blood pressure control, and that should be thought of in terms of local control and systemic control, and in the short term and the long term. When a person is experiencing a tissue injury and there is changes to blood vessels in the area of injury, which includes dilation of vessels, that should not impact the actual systemic blood pressure unless it's a huge traumatic injury with lots of blood loss. Those small changes to an area of cellulitis that occur at those blood vessels should not translate into a complete change in the systemic blood pressure. So some of the local mechanisms are only to meet the needs of the body at the time. Local control of blood pressure, nutritional needs of tissue, and reaction is related to the endothelial cells. And many of these factors we have talked about. So endothelial cells are quite busy. They are responsive to and make many types of substances. Endothelial cells are able to make endothelium 1 and nitric oxide to control their diameter, and they are responsive to histamine and prostaglandins and bradykinins, and these are the things in this area that are part of the inflammatory process. Serotonin too, but we didn't talk about that one as much, are all ways that the vessels locally can dilate in order to meet the needs, especially in times of injury or inflammation. Short-term systemic blood control is managed by the autonomic nervous system, which is the involuntary part of our nervous system. When we talked about the central and peripheral nervous system last class, we then divided the peripheral nervous system into the afferent and efferent system, where the afferent system was picking up sensory environmental information and sending that to the brain for interpretation. And then the efferent system was sending information out so that the body could do something based on that information. So there was the voluntary side where we had muscle planning movement strategies where we're moving our muscles. And then there was the involuntary side, which is the autonomic control of our body based on what's going on around us. and that's the sympathetic nervous system and the parasympathetic nervous system. The sympathetic nervous system has a lot of moment to moment control of our blood pressure and will respond very quickly moment to moment changes in our environment, like when we change positions. That's one of the times that our autonomic nervous system is in play to help us maintain our blood pressure. The cardiovascular or vasomotor centers in the brain are located in the medulla. There was the part of the medulla where the somatic nerves crossed over, and then there was a part of the medulla that was in charge of basic functions, especially related to our vital signs, and that output controls blood pressure and heart rate. The sympathetic nervous system, when that is activated, that increases the output of epinephrine and norepinephrine, which will affect our blood vessels and our heart. If we switch to the parasympathetic nervous system, then the output changes, and there's a decrease in epinephrine and norepinephrine. So we always think of the sympathetic nervous system as being the fight or flight response. I always like to remind people that our sources of stress, because it's tied to the stress response, this is all developed for the evolutionary prehistoric person, and our sources of stress at the time of our development as Homo sapiens was starvation, dehydration, trauma. That was our stress. So our body's sympathetic nervous system response is really perfect for those types of stressful situations. It is not a good response or perfect response for our current stressful situations, including, work, bills, class, this class, the things that we have to deal with now in our relatively pampered experience. So we have to remember that when we have increased stress now, it's actually not very adaptive like it was before. Right. So the sympathetic nervous system is releasing epinephrine and norepinephrine. That's this sort of control center is located in the medulla, and these neurotransmitters will travel in the bloodstream to the receptors in the body that they work on. And the sympathetic nervous system receptors are alpha receptors and beta receptors. Alpha receptors are located on peripheral vessels, just one of their locations, and beta receptors, there are many locations, but in terms of this, are located on the heart. If epinephrine and norepinephrine stimulate alpha receptors on blood vessels, there's an increase in vasoconstriction because there's contraction of the smooth muscle of the vessels, which means we decrease the radius, which is the biggest influence on resistance in general. And we know that an increase in resistance will decrease blood flow. But the reason the body does that in response to stress is because if I vasoconstrict peripherally, then I shunt blood to the core, especially the brain, because the brain has no reserve. That's what we learned last class, that it requires a lot of glucose and a lot of our cardiac output, but it cannot store oxygen or glucose, so it's extremely sensitive. So our body will sacrifice for the brain. If epinephrine and norepinephrine stimulate beta receptors on the heart, then the heart rate increases, and the strength of contraction increases so that we can increase cardiac output. Because obviously we might need more blood volume and oxygen to feed the demands of our body under stress. Whereas the opposite would occur during parasympathetic nervous system stimulation. There's still a section of the medulla that does increase output of the parasympathetic nervous system, and that is acetylcholine that's released. The parasympathetic nervous system is rest and digest. Sometimes people think that means everything slows down, but really it's just a change in priority. Now that the action is over, we got to finally eat and rest our bodies. It's now time that we can take care of the other needs of our body, which is getting nutrients from food and elimination of waste. And that's the parasympathetic nervous system is much more active than our GI and GU tract can do their jobs. So we don't need those blood vessels to be very constricted because then we can't get blood flow to our digestive tract. So in this case, the vagus nerve, which is one of the main nerves coming from the medulla, that acetylcholine will cause vasodilation and will decrease heart rate because our priorities are different essentially. So that is what this picture is showing you, that vagus stimulation will cause a decrease in heart rate and strength of contraction, whereas sympathetic stimulation and vagus stimulation will try to calm down the sympathetic nervous system, and sympathetic nervous stimulation will increase vasoconstriction and increase heart rate and contraction. So that is happening all the time. Obviously, it could be because of large things that are happening, but also just changes in our position. If I stand up, then gravity means that my blood would be in my legs instead of in my heart and brain. And that obviously cannot be, or we pass out. So the sympathetic nervous system is very, very sensitive to changes in the amount of blood that is near the heart because of what are called baroreceptors. So baroreceptors are located near, you can see here, near the carotid sinuses and the aortic arch. So right by our neck and right where the aorta comes off, so those large vessels, they're going to notice if the pressure is different. They're baroreceptors, so they're responding to stretch. If these vessels are stretched out, then there's enough volume to support blood flow to the brain, and we are not, then there is less sympathetic nervous system output because there's adequate blood flow to the brain. But if the person stands up, say the person stands up and now that gravity has pulled more of the fluid into the legs, then there is some collapsing of these vessels. There's less stretch. That's a trigger. Less stretch is a trigger to increase sympathetic nervous system output in order to cause vasoconstriction and a potential increase in heart rate to then right this, to keep the blood volume up towards the head, and those vessels will again stretch to where they're supposed to be, and that output will decrease. So it's kind of a negative feedback system responding to our position change. So that's helpful. The other receptors that help to regulate our blood pressure from moment to moment based on our needs are chemoreceptors that are located in arteries. So these chemoreceptors are located in the arteries that are near the medulla. And they're going to respond to an increase in carbon dioxide and a decrease in pH. This means that there has been an increase in metabolism or metabolic needs. Because if we have an increase in carbon dioxide and a decrease in pH, that means we have an increase in metabolic waste products. So we need to increase blood flow if possible. So we need to increase heart rate and strength of contraction is the whole point. The thought is, okay, we're under stress, so we're going to need to increase that output. So that's short-term control, moment to moment, based on stress in the body and just moment to moment changes based on metabolic demand and position change. And that's one of the large jobs of the autonomic nervous system in maintaining our heart rate and blood pressure. The other more long-term management of our blood pressure is done by the kidneys, by a special system called the renin-angiotensin-aldosterone system. And that might hopefully vaguely sound familiar as because it's the basis of a lot of blood pressure medications that you might have given people many times, especially like lisinopril or spironolactone or things like that. It's based on a special area in the kidney. That's where it starts. So that's a kind of a different position of the nephron than people commonly think. So here's my nephron with Bowman's capsule. So inside the nephron, that's very sensitive to pressure changes in the body. There's that glomerulus, which is the blood vessel that interacts with Bowman's capsule of the nephron, because this is where we would then filter fluid that would either come out as urine or we would reabsorb. There is a big pressure monitor here. What is the pressure in the glomerulus? That monitor is called the juxtaglomerular apparatus. It's monitoring very closely what the pressure in the glomerulus is, because that is going to indicate to us potentially what blood flow is. So now again, remind yourself that the reason the system was developed, it was adaptive for prehistoric people. If there's less pressure coming into the glomerulus, it's usually related to a decrease in fluid, which means we're dehydrated, starved, or have suffered massive trauma. So we've lost blood. So the body's interpretation, or the reason this system developed in terms of evolution, the survivors had this system because they could detect fluid loss with great sensitivity in the kidneys and then adjust by this hormonal cascade that we're going to talk about. However, that is not the reason. I mean, sometimes it is if we're dehydrated or whatever, or are suffering from trauma-related issues. But many times this system is overactive because there is a sense of decreased pressure in the glomerulus. But that's because of decreased blood flow. And the reason there's a decrease in blood flow is because there's an increase in resistance related to hypertension itself. And so this can become a vicious cycle. Someone developed hypertension for whatever reason that is, which we'll talk about later in this class. So hypertension is developed, and because hypertension increases resistance, there will be a decrease of blood flow to our end organs, including the kidney. And the kidney will interpret this decreased flow as a reason to activate the renin-angiotensin-aldosterone system, which will further increase blood pressure. So keep that in mind, because this is a great thing. This does work really, really well to bring blood pressure up, especially in times when we have fluid loss. However, it's not helpful if that's not the reason why it got activated. So these are the steps we go through. So some of this might sound familiar. So the juxtaglomerular cells are sensing a decrease in pressure related to what is thought to be a decrease in blood volume related to hemorrhage, dehydration, something like that. When that happens, when the juxtaglomerular cells sense a decrease in blood volume, a decrease in the pressure inside the glomerulus, those specialized cells will release an enzyme called renin or renin, whatever you would like to call it. This is an enzyme that will convert angiotensinogen to, because of that action, angiotensin 1. So angiotensinogen is an inactive protein that is created by the liver that's floating in the bloodstream waiting to be needed. It's like if you need me, I'm here. I won't do anything until you show you need me. Renin will convert through an enzyme reaction angiotensinogen to angiotensin 1. Angiotensin 1 will be converted to angiotensin 2 by angiotensin converting enzyme. This transformation occurs in the lungs after blood returns to the heart and gets pumped out through the right ventricle into the lungs. The angiotensin 1 that is circulating will interact with angiotensin converting enzyme in the blood vessels of the lungs, and that will convert angiotensin 1 to angiotensin 2. So at that time that oxygenated blood comes back to the left side of the heart, there'll also be a large amount of angiotensin 2 that's going to be pumped out into the general circulation. When it does, that angiotensin 2 has two actions. It is a powerful vasoconstrictor. That's one of its big actions. And so it'll constrict the smooth muscles of arteries, causing a decrease in radius, which increases resistance. And that'll increase blood pressure. And it will activate receptors on the adrenal gland that will increase the release of a hormone called aldosterone, which saves water in the nephron, in the kidney. So then we increase volume. So if this was all activated because of dehydration or hemorrhage, then these steps lead us to increase in peripheral vascular resistance and an increase in fluid volume, which would bring our blood pressure up. So it would be an excellent way to bring our blood pressure up if that's why we needed it. However, like I said, sometimes this becomes activated because of chronic hypertension. And this is why this is one of the first line drug targets, is turning the system off. Because if I can give somebody an angiotensin converting enzyme inhibitor or an ACE inhibitor, then I can stop this vicious cycle, which of course you'll learn more about in pharmacology. So that is our blood pressure mechanisms. Does anyone have any questions about mechanisms of blood pressure control? I know, isn't it? It's very cool. Okay, so we're moving on to the heart. So some of this stuff should obviously be reviewed. Again, the anatomy of the heart, there's the outer fibrous covering to keep it in its fixed position and protect it. In its position in the chest, the myocardium is the muscle portion, and the endocardium is the inner kind of slippery layer of the lining of the heart. There are valves in the heart that are supported by its fibrous skeleton. So now here's my heart for you guys. Which blood vessel empties into the right atrium? passive um, filling of the right atrium comes from the superior. Yep, superior and inferior vena cava passively flow constantly into the right atrium. And then, so that's coming from both directions, and then blood will flow through the right atrium into the right ventricle through which valve? The tricuspid, Erica, that's right. And then it'll get pumped out from the right. Oh, great, Karis. You're like rocking this. So there, through the tricuspid into the right ventricle and then out which vessel? Pulmonary artery, right. So we're, the pulmonary, that's the only artery with deoxygenated blood and it goes to the lungs. So there's our pulmonary circulation. That then empties once the deoxygenation occurs. Through the pulmonic semilunar valve. We then we go through the lungs, we have oxygen exchange and carbon dioxide exchange, and then the pulmonary veins empty into the left atria, and the blood goes to the left ventricle through which valve? It's a race between Erica and Karis. The mitral valve or the bicuspid. It depends on how you learned. I always said mitral. It's supposed to be the mitral valve is a Catholic reference to the Pope's head that looks like a mitre. I don't know. This random trivia for you for no reason. And then out the aortic semilunar valve into the general circulation. Okay, so that's the way the blood flows. Now you're going to have to take me through the conduction system. I have poor drawing here. This is the P, QRS, and T wave. What does the P wave signify? Oops, I'm hitting the wrong button. So the P wave is depolarization of the atria. Yes, depolarization. So you can never have a muscle contraction without electricity. So the electricity always comes first. If this is my heart, I either draw hearts as boxes or eggs. Here's my SA node, which is the pacemaker of the heart, the sinoatrial node. At this point in the P wave, as we go through the whole thing, the entire atria depolarize through these intranodal tracts to the AV node. There's a pause here after complete atrial depolarization that allows for atrial contraction, the last part of diastole, which is the atrial kick. And then we get to the QRS. So what's happening during the QRS? Correct, ventricular depolarization. So you'll notice that the wave is larger, but not yet contraction, because contraction occurs after electricity. So you'll notice that the QRS is larger and faster, and that's because there's a lot more surface area to the ventricle, and there's a lot more conduction, specialized conduction tissue to the ventricle. So there is more electrical activity that occurs. So we start at the AV node, we get to the bundle of His, then we get the separation of the bundle branches, and then each cardiac myocyte is fed by a different Purkinje fiber that is branched off the bundle. And so each ventricular myocyte is given electrical stimulation from the Purkinje fibers. So this is our QRS depolarization, and then here is ventricular contraction or systole. So what is the T wave? Ventricular repolarization, correct. So then the ventricles go back to resting potential. Where is the atrial repolarization? Because they have to do that too. Yes, it's buried in the QRS. You can't see it because the electrical activity is so much smaller than the ventricle, so it gets lost in there. Okay, so this is our ventricular contraction, and this is our atrial contraction. Atrial contraction is the end of diastole, and this, of course, is systole, and that's the beginning of diastole. So if I had, here's the P, QRS, T, P, QRS, T, P. If I did that, okay, here we go. So this is systole, and this is diastole. So obviously we are in diastole longer than systole because we need more time to fill than we need to eject volume out into the system. So then when it's diastole, which valve is open and which valve is closed? So during diastole, the atrias will be open because we want to be able to fill. We want to be able to fill the atria to the ventricle because that's the filling time. So the atrioventricular valves will be open and the semilunar valves will be closed. And then the reverse occurs in systole. We want those atrioventricular valves to be closed and the semilunar valves open so that when we eject volume out into the vessel will be able to accept it. So this is the period between S1 and S2. Because S1 to S2 is, and then the next one, S1, this is systole between here, and this is diastole. So when you're hearing the heart noise, you're hearing the valves close. So S1, you're hearing the atrioventricular valves close, and S2, you're hearing the semilunar valves close. So that's important, especially when you get to physical assessment, because you can start to distinguish if you hear extra sounds in between, if they're part of the systolic or diastolic phase of the cardiac cycle, and then what valves are implicated, etc. So here you go. We talked about the conduction process. That is how the SA node is influenced by the sympathetic nervous system. That's how we can control the rate, which we mentioned. We talked about that. We talked about that. So we talked about the cardiac cycle, divided into two parts. Systole is when the ventricles are contracting, and diastole is when they are relaxed to fill. And at those times, during diastole, the semilunar valves will be closed and the AV valves will be open. And then during systole, um, the semilunar valves will be open and the AV valves will be closed. That's such a weird looking picture, but that is how the valves should be during the cardiac cycle, and then that's what you're hearing when you listen. The coronary arteries are branching off actually near the aortic semilunar valve. You can kind of see that. That's where the coronary arteries branch off to feed the right and left side of the heart. The coronary arteries are fed when ventricular contraction takes place and the valve is open. There is showing you blood flow going through into the system. During ventricular